Hello and welcome everyone to this podcast for human genetics. In this podcast, we're going to talk about a few different things, including human development, more precisely development of the embryo and into the fetus. We'll talk a little bit about twins and the effect of teratogens. But we're going to begin with talking about human development. And as I said, we're going to focus primarily on embryonic development. Now, we've talked a little bit about embryonic development earlier when we were talking about stem cells. From the last podcast, we talked about how the egg was formed through meiosis, and then how the sperm was formed. Here we're going to pick that story back up again and talk about how when the sperm fertilizes the egg, that we form this single-celled zygote. I drew this bigger than I should have because the zygote is really no bigger than the egg that was used to make the zygote. But I'm going to need a little bit of space to draw inside of the zygote as it develops through embryogenesis. Okay, so they form this zygote. Remember in our discussion with egg development in the last podcast, we talked about how there was a meiotic arrest during its development. And once it's fertilized, this is when the embryo can now re-enter meiosis, and then it'll start meiosis 2 after it's been fertilized. The cell will divide several times here, and at this division here that I'm going to show you, there's going to be 16 cells. We call this stage the morula stage. And this is day 4. And remember the zygote, this is going to mark day 1. The next thing that's going to form, and we mentioned this in the previous podcast, but let's go ahead and draw it again. I'm going to draw it slightly differently. There will be this outer portion of this stage of cells, and inside of it will be more cells. And I'll draw them in a slightly different color here. This stage is called the blastocyst. Well, the structure is called the blastocyst. This forms around day 5. These green cells on the inside, just to remind you, remember those are our inner cell mass. These are what will ultimately become the body cells of the developing embryo. These are, remember, the embryonic stem cells. They are pluripotent. And I'm going to extend our drawing here to the bottom row here. And I'm going to pick it up here. And this is still at the blastocyst stage. But after the blastocyst forms, shortly after that, it will implant on the uterine wall, still with that inner cell mass shown here. So let's just write implantation. And that usually occurs around day 7 or so. Now a few days will pass. In fact, it's not until towards the end of the second week and beginning of that third week where we have our next stage of embryonic development. And this stage is called gastrulation. What this word actually means is the formation of the gut. And we'll talk about why it's called that in just a moment. Now my artistic skills will be challenged here in trying to draw this, but I think we can make it work. What happens with this inner cell mass is those cells will flatten out. And I'm going to draw the cells in different colors just to distinguish them. Because as they flatten out, they begin to specialize. That is, they begin to lose their pluripotent state. These cells here will specialize to form a layer of tissue that we call the ectoderm. These cells up here will specialize to form the endoderm. And what forms in between these, sort of like a sandwich, are a different set of cells that are called the mesoderm cells. I've heard them called mesoderm and mesoderm. I'll probably alternate between the two. Again, as I said, these now have specialized. They don't have the same state as these cells here. They can no longer become any type of body cell. They have some limitations placed on them. And I don't know if I mentioned it, but let me make sure I write it down here. This forms towards the end of week 2 and the beginning of week 3. And then throughout the rest of embryonic development for the next several weeks, what will happen is these tissues will begin to form important body structures. And so I'm going to list some of those here. The endoderm will ultimately become the digestive tract. It'll also form the liver and the pancreas. The mesoderm will form muscles, blood, and blood vessels. The ectoderm will form skin. It'll form the nervous tissues and sensory organs, such as the eyes and the nose and the ears. I've only listed a subset of the tissues they ultimately become, and these are the only ones you need to know for the purpose of this class. But there are other tissues that form, and you can look those up. There's a few others listed in the book, but this is all you need to know as far as what these 3 tissues ultimately become. Now, as I said, as this embryo develops, these tissues will form, but something else happens as well. As this embryo is implanted on the wall, some additional tissues need to form that will support this developing embryo. So I want to talk about those supportive tissues next. As we describe some of these supportive structures, I want to refer to this picture here. As you know, my drawing skills are limited, and the first supportive structure I want to talk about is something called the chorionic villi. The chorionic villi, these are structures that extend from the embryo into maternal blood. There isn't mixing of the blood between the developing embryo or fetus and mom, but the chorionic villi brings in some of the blood vessels from the embryo, and it is able to absorb nutrients from this pool of the maternal blood. So nutrients can move across. Waste from the embryo can move out of the embryo. And the other thing that can move from the embryo into the maternal blood is cell-free DNA. So cells don't move from the embryo across the blood vessels into mom, but any DNA that has been lysed or lost from the embryo cells, that DNA can move across. The next supportive structure I want to talk about is the placenta. So what happens is that as the embryo matures, the chorionic villi becomes the placenta. So around 10 weeks of embryonic development, the placenta forms. It is much larger than the chorionic villi by themselves, so it can handle much more nutrient transfer, and it also will begin to secrete hormones as well. The next 2 structures I want to talk about are structures that in non-humans have a little bit more of an importance. They still play a role in human development. The first is something we call the yolk sac. It is what you may think it is. For instance, in chicken eggs, it is the yolk that we eat. And it provides nutrient for the growing chicken. Humans don't need it for the nutrients, but it still serves some function. And during early embryonic development, blood cells form here in the yolk sac. And over here in the picture, you can see where the yolk sac is, in close proximity to the chorionic villi. The next structure I want to talk about is something called the allantois. The allantois has a few functions that still exist in human development. It helps provide nutrients, and it will give rise to the umbilical cord. So the next structure I want to talk about is the umbilical cord. And you can see the umbilical cord over here in this picture, extending from the embryo, developing embryo, into what's shown here, the chorionic villi, but it will extend into this as it forms the placenta. So umbilical cord. And I'm just going to write here connects the embryo and later the fetus to the placenta. And the last structure I want to talk about that provides support for the growing embryo is the amniotic sac. And you can see the amniotic sac. It's this fluid-filled cavity here that surrounds the embryo. So it's filled with amniotic fluid. It contains waste products from the developing embryo or fetus. It also has fetal cells in it, cells that have been stripped away from the fetus as it's growing. The amniotic sac also protects the embryo and ultimately protects the fetus. It serves as a shock absorber of sorts. So these are all important structures that help protect the developing embryo and fetus. But we can use some of these structures to check on the relative health of the developing embryo or fetus. So let's talk about how we can do that on the next whiteboard. There are 3 prenatal tests that can be done to check on the health of the developing embryo or fetus. The first is chorionic villi sampling. The second is amniocentesis. And the third, which isn't shown in this picture up here, is maternal blood sampling. So let's go ahead and talk about these. With chorionic villi sampling, a tube is placed into the uterus and it removes cells from the chorionic villi. These can be taken from the developing embryo as early as 10 weeks. With amniocentesis, a needle is placed through the abdomen into the amniotic sac to withdraw some fluid from the amniotic fluid there. Remember, there are going to be some cells that are removed from the developing fetus that are going to reside here in the amniotic sac, so they can be analyzed. So this can happen as early as 15 to 20 weeks. So an advantage of chorionic villi sampling is that you can do it sooner. Another advantage of chorionic villi sampling is that it is quicker to process the cells. So I'm going to put quicker processing. With amniocentesis, you process the cells in a similar way, but it takes a little longer. So it's not as fast, and you have to wait longer. Both of these, the risk of both of these is that there are chances of causing a spontaneous abortion or infection, or in the case with amniocentesis, very rarely does this occur, is that you could cause injury with the needle. Now, with maternal blood sampling, it has a few advantages over the first two here. Remember when I told you that the chorion forms here and later it becomes the placenta, and we have nutrients that go between mom and the developing embryo or fetus. And I said that cells can't go across that barrier and enter a mom's bloodstream, but any DNA that has been lost from cells, that DNA can move across that barrier, and the DNA, the free DNA from this embryo, can now be found in the mom's blood supply. So maternal blood sampling can be done as early as 8 weeks of development. And we know at about that stage, about 10% of all the free DNA, that is DNA not found in cells, in mom's blood supply, is of embryo or fetus origin. Having the DNA, you're not going to be able to build a karyotype profile like you can with the first two, but you can screen the DNA for various characteristics that can lead to an understanding of what kind of diseases the embryo may have. Okay, the next thing I'd like to talk about. All right, the next thing I'd like to talk about are twins. There are three types of twins I would like to talk about. The first are what we call dizygotic. These are commonly referred to as fraternal. The other type of twins I'd like to talk about are monozygotic. And these are commonly referred to as identical. And then the third type are conjoined twins, sometimes referred to as Siamese twins. So dizygotic twins form because two separate eggs, so two different eggs, are fertilized. So two separate eggs, two separate sperm, and so that's why we call it dizygotic. Di for two, and two separate zygotes are formed. These two zygotes that form, as far as the genetics are concerned, are no different than two other siblings. Ultimately, they will share that same prenatal environment, and that in itself makes them unique. But as far as the genetic level is concerned, they're the same as any two siblings. Now, there are several kinds of monozygotic twins that we could talk about, but mainly what I want you to know is this, that there is a single egg that is fertilized by a single sperm. Mono for one zygote is formed. What happens to this fertilized egg, it will begin to develop as a single embryo. And then what happens, sometime during early development, it will split. And you will begin to form two separate embryos. If it happens sometime roughly between days 1 and 12, you will end up with a monozygotic twin. You should know that if it happens between days 13 and 15, you will end up with a conjoined twin. So, conjoined twins, again, a single egg is fertilized. It will divide as a single embryo for a certain period of time. But by the time it gets to, so let's draw some more cell divisions here. So by the time it gets to days 13 to 15, if it divides after that into two separate embryos, what will happen is they will be conjoined. And for the purpose of this drawing here, we'll draw them conjoined right here. Now these cells will continue to divide, and each one here will form the blastocyst, and then each one will form the gastrula to form the different tissues, and each individual embryo here will form independently, though attached. Now, monozygotic twins, they will also, of course, form independently of each other, but they're not connected with each other. It seems unlikely I would ask you on a test what's the chances of this happening, but you might just be interested. It's typically somewhere between 1 and 50,000 to 100,000. So it's pretty rare that this would happen. Monozygotic twins, I might as well tell you, this can happen 3 to 4 pregnancies per 1,000. So it's more common. However, in about 70% of the cases when monozygotic twins form, one of the twins stops growing and only one of them survives. Your book refers to that as the vanishing twin. Dizygotic twins happen much more frequently. So I'm just going to put more frequent. In fact, we see more of these now because when we use artificial reproductive technologies to help couples have children, often a result is multiple pregnancies due to the dizygotic twins. I should also mention that you often hear people say that twins run in families or they skip generations and things of that nature. And from what we can tell, the types of twins that run in families, so to speak, are the dizygotic twins. So this is heritable. And the reason it's heritable is that there are various mutations a woman could have that would cause her to produce more than one egg a month. And if she produces more than one egg a month, she is more likely to have dizygotic twins form, as opposed to somebody who just usually produces one egg a month. There's less evidence that monozygotic twins or conjoined twins have a genetic component or heritable component to them. All right, the last thing I want to talk about are teratogens. And so let's give it a quick definition first. These are various agents. They could be chemicals. In fact, often they are chemicals that cause embryos to have birth defects. And I want to briefly introduce 4 of these to you. The first one is thalidomide. Thalidomide was actually given to women as a treatment for morning sickness. And it was fairly effective at treating morning sickness. Now, unfortunately, at this time, many doctors did not think thalidomide or any medicine could really go through the placenta. Unfortunately, they were wrong, and this led to many limb malformations. Some women might take this for morning sickness early during pregnancy because that's when morning sickness is the worst, but that is also the time that the limbs form. This was primarily seen in Europe because in the United States it still had not been approved, and there were studies that had shown that thalidomide caused birth defects in test animals. So it was never prescribed in the United States. It's prescribed now in the United States and elsewhere in the world to treat as a treatment for AIDS and certain kinds of cancers, but not used anymore to treat morning sickness. Alcohol is another teratogen I'd like to talk about. We know that women who drink alcohol, even small amounts, risk the chance of their baby being born with fetal alcohol syndrome. Fetal alcohol syndrome can have various effects on the child that is born. They can have behavioral problems, cognitive, learning problems, communication problems, an increased chance of having ADHD, and various other potential problems. It's important to remember that early in development, when the nervous tissue starts to form, and then throughout embryonic and fetal development, as the brain and its tissues continue to develop, alcohol can have an effect. The next one I want to write down here is nutrients. This may sound a little weird to say that nutrients can serve as a teratogen, but in excess amounts, some nutrients will have the potential to lead to birth defects. So some examples include vitamin A. Vitamin A or a derivative of vitamin A can be found in various medicines for acne or psoriasis. These can lead to spontaneous abortions or various defects like heart defects. And linked to nutrients, of course, we would say the lack of nutrients or malnutrition can lead to a smaller placenta, a smaller baby, and I'm running out of room here, so I might just indicate one other things, and it can lead to learning problems. The last one I want to talk about are viruses. Not every virus is a teratogen, but some are. And the reason that viruses can be a problem is that they are small enough to cross the placenta. The chickenpox virus can be devastating for a developing fetus. Zika virus, and this is often found in sperm, and so after pregnancy, if a man has Zika virus, he could pass that on to the developing baby, and of course to the mother. HIV can be passed on to the developing embryo or fetus, And the last one I want to talk about is the German measles virus, also known as rubella. And this is one that can lead to deafness. The last thing I want to talk about is something called the critical period. And this is the time period that certain structures of a developing embryo or fetus are most sensitive. For instance, when we talked about thalidomide, we know that limbs and digits, there's a relatively short period of time that they can be affected early during embryonic development. After that, drugs that affect limbs and digit development are less of a problem. However, things like the brain, they have a much longer critical period. Pretty much throughout the entire pregnancy, anything that affects brain development is a problem. That's why alcohol at any point during pregnancy can lead to many defects such as the fetal alcohol syndrome. Though any time during pregnancy can create problems, we know that embryonic development is the most sensitive. And the scary thing about that, I've always thought, is that sometimes women don't know they're pregnant during this very critical time early in the gestation period. I should also point out that embryonic development, that time period is about the first 10 weeks from fertilization on. All right, that is all I have for this podcast. In this podcast, we talked about embryonic and fetal development. We talked about how twins are formed, and we talked about birth defects. If you have any questions at all, please make sure you reach out to me and I'll be happy to answer any questions you bring to me, or you may want to ask members of your team. But that's all for now. Have a good one, and I'll talk with you later. Bye.